Hello, you are most welcome, ladies and gentlemen, to Real Impartation Moment on Tuesday Night Anatomy with Daniel Okwan. Today's section, we continue our journey of the integumentary system, and there's a second part, which I've tagged the epidermis. And so, without much ado, let's set the ball rolling. So, we've actually seen that, yes, the outermost, or let me say the outer component of what we call the skin is the epidermis. And that's the point where frequently some people will bear that kind of hair. You see that the sebation, excretory ducts of course, the sweat glands will of course open over there, so you have that kind of pulse over there. So that is the epidermis. Very important. So then, what is this epidermis you are talking about? Now remember, the epidermis, coming from the epidermis, you can derive all these appendages of the skin, like the sebaceous glands, like the sweat glands, like the hair follicles. Although they grow, can you see that they grow towards the connective tissue area? Now, for you to know that you've gone beyond the epidermis, then there will be that kind of junction over here, which of course separating them will be the basement membrane. So this is the epithelium, the epidermis, and beneath is the connective tissue dermis. So over here will be the basement membrane, but you see that the connective tissue will be moving into the epithelium, and they are what we call the dermal papillae or the connective tissue papillae, while the epithelium will also be dipping into, of course, the connective tissue. That is what we call the epidermis. So once you can see these things, which of course we know that under hematology eosin staining or any other stain, we tend to see that yes, the epidermis tends to pick up the stain quite intensely than what we call the dermis. So that's what we tend to see. So beginning with the deepest layer of the epidermis, and that's that layer. Okay, so beneath that layer, you have what you call the papillary layer of the dermis. The deepest layer of the dermis is known as what we call stratum germinativum. And this stratum germinativum really consists of about 1 to 2 layers of cells, and they tend to be, these kind of columnar shaped, cells. That's stratum germinativum. And one thing that we find frequently in the stratum germinativum is that we tend to see mitotic figures, because over there in the stratum germinativum, that is that layer which is going to be the stem cells. So it's undergoes mitosis to replace these cells in the subsequent upper layers of the epidermis. Now, once we get these cells moving into, of course, the next layer, then they begin keratinization. They begin to, build up some kind of keratin in the cells. Now, I also show you something that we find in the stratum germinativum, and that one will going to be the melanocyte. I will show you that in our next, meeting. So this is what we call these cells bearing these kind of columnar shaped cells. That is the germinative layer, stratum germinativum, or the stratum basale. Okay, so once you have something like some kind of bends and it affects what we call the germinative layer, then it means that the epidermis cannot grow again. And that is why frequently people are advised not to be putting harsh substances on their skin, because once it bends all these layers and it reaches and it bends completely the germinative layer, then it means that yes, of course, you'll be prone to whatever thing. Then of course plastic surgery becomes the option, because what will happen is that the epidermis, which is going to protect the dermis, yes, everything, yes, will not be able to grow again. But the growth of this epidermis is actually highly, influenced. Okay, it's actually highly influenced by what we call the epidermal growth factor, and these epidermal growth factor are actually produced by some glands, notably the salivary and of course the duodenal glands. Okay, so that yes, what will happen is that it will stimulate, these germinative cells or the basal cells of the epidermis, and it also accelerate keratin production, because as they are produced from there, then keratinocytes, these cells become keratinocytes and they will be producing what we call keratin. Then they also stimulate epidermal repair, because for repair to take place, what will happen is that these basal cells will have to proliferate, will have to undergo this kind of production to replace the uppermost cells. So for instance, we get to know that as the squamization takes place in what we call the stratum corneum, then what will happen is that yes, the epidermal growth factors will stimulate what we call the basal cells to proliferate, and to also stimulate glandular secretion. Glandular activity will also be there. So what have we seen? We've seen that made up of about 1 or 2 layers. Yes, mainly 1 two layers. We tend to see what we call the stratum basalis. We've seen that that is the deepest layer, yes, and it's also be attached, okay, or to be connected to, of course, the underlying papillary layer of the dermis via a basement membrane. We've seen that. Then we see that it will have some other cells, and those cells notably will be, of course, the melanocyte, apart from having the stem cells in there. Then also frequently we see that kind of Merkel cells over there. We will see that. Now, moving away, so once these ones proliferate, they will give rise to the next layer, and the next layer, yes, that's what we call the stratum, spinosum, stratum spinosum. And for stratum spinosum, what we find is that they tend to have that kind of spiny, processes. Some kind of processes, arise from these areas. Now, it is these sites that we call these cells actually keratinocytes, because they start, producing what we call, keratin as a protein. Now, one key thing that we should also be aware of that is that these keratinocytes will be connected via, some kind of cell junctions, and these ones will notably be what we call desmosomes. Okay. Now, frequently we also see what we call Langerhans cells. Langerhans cells will be found in the stratum spinosum. But sometimes you may also see some melanocytes moving from the stratum basale into, of course, the stratum spinosum. Okay, so that is what I want you to actually know. Then we go to the next layer. So now one thing that we have to know is that these cells will now begin to contain what we call some kind of granules, notably keratohalin granules. Okay, keratohalin granules in what we call the stratum stratum granulosum. So it makes it granular. It has a granular, those cells will tend to have granular cytoplasm, okay, and because of the keratohalin granules in there. Okay, and that one, yes, usually will be made up of 3 to 5 layers of cells. Stratum granulosum will be made up of usually 3 to 5 layers of cells. Now, one thing is that upon migration, they will go to be in the stratum lucidum, and then when stratum spinosum, yes, can also give rise to, of course, stratum granulosum. So it's going to be in that order. So that's how the layers will be forming. Stratum basalis, yes, will undergo mitotic activity to give rise to these keratinocytes, okay, which will bear what you call processes, okay, which we call, stratum spinosum, which will be connected by, of course, desmosomes. And then frequently also in the stratum spinosum, we will see what we call these Langerhans cells, and then sometimes melanocytes will also be seen there. Then in the stratum granulosum, it will tend to have granular cytoplasm having 2 to 3 layers of cells, and that one, the granular layer is due to the presence of what we call keratohalin granules. Okay, so that is what we see over here. And what is happening is that these gradually the cell membranes thicken, okay, and the organelles disintegrate and the cells tend to die. So this is the area where the cells begin to die because they contain a lot of keratohalin granule. Because of a lot of keratin in there, then what happens is that the cells begin to die. Because of the presence of keratin, the cells die. And then we go to the next layer, a clear layer, which will be made up of dead cells. Actually, that is what we call the stratum lucidum. Now, for stratum lucidum, usually it's about 2 to 3 layers of cells. The cells will be dead because they will start dying in the stratum granulosum because of the keratohalin granules over there. But one thing that we tend to see, this lucid or clear layer, we tend to see it's almost always in thick skin. But in thin skin, yes, we don't tend to see it. We don't tend to see it in thin skin. Then beyond that, we go to what we call the stratum corneum, and the stratum corneum is actually the thickest layer, consisting of about 20 to 30 layers of dead cells actually. So those cells are actually flattened dead cells. They don't have any nuclei. Yes, they are actually dead cells, and it's very important in making the epidermis water resistant. Okay, water resistant, but not waterproof, actually water resistant. Okay, so that is what we see, and it also permits slow water loss by insensible perspiration in the stratum corneum. So the stratum corneum, you may also call it the horny layer. Yes, stratum lucidum, that's the clear layer, stratum granulosum, the granular layer, then of course stratum spinosum, the spiny or the spinous layer, and then stratum basalis, the basal layer. So these are the layers of the epidermis of the skin. But one other thing too happens, okay? And because what happens is that in the granular layer, we see certain things in what we call the keratinocytes over there, because most of the cells of the epidermis of the skin, about 90% of them are actually keratinocytes. So in the granular layer, filaggrin, okay, there's a protein which we formed that from profilaggrin. We call it filaggrin. And that one will be rich in a lot of histidine amino acid. Even apart from that, that kind of glutamic acid. Okay, apart from histidine, it also have that kind of glutamic acid. Then it is highly phosphorylated. Okay, so it's highly phosphorylated. The protein is called filaggrin, but it is made initially from the pro form, which we call it the profilaggrin, the inactive form, and it will contain a lot of histidine and as well as glutamic acid. Then it is highly phosphorylated. Now, what happens is that they tend to aggregate keratin filaments and flatten what we call the keratinocyte. So we said that in the granular layer, that is where the cells begin to die, because a lot of these keratin will be aggregated by these filaggrin proteins, okay, so that they tend to flatten even the keratinocyte. And that is why as you move apically, the cells tend to flatten as you move apically. When they die, their nuclei become pygnotic and of course eventually gets lost. By the time you have a lot of keratin in there with, pygnotic nuclei, which, they've not lost it anyway, that becomes parakeratinization. But eventually they become, they actually they disintegrate, the nuclei disintegrate. Now, one other thing too that you have to know is that other events, okay, include the release of lipids and cell envelope proteins from what we call Golgi-derived, organelles, Golgi apparatus, and these ones are known as what we call laminar granules, okay, which will form a very important skin barrier. So in the granular layer, in what we call the filaggrin, apart from we forming the filaggrin, other things which happen in the granular layer is that we form these kind of laminar granules in there, which are very important in becoming a very important skin barrier. Now, once these cells migrate, okay, from the granular layer into what we call the corneal layer or the horny layer, then what will happen is that there's going to be what we call proteolysis of these filaggrin proteins. So they break down, okay, and when they break down, they are going to release, of course, this histidine that we said, because we said that these filaggrin will be rich in protein, will be rich in, of course, histidine, even glutamic acid, and of course it will be highly phosphorylated. When that happens, it's an amino acid, so this kind of histidine, when it breaks down, then it will be deaminated. The amino group will be taken out, and that one will give us what we call trans-uroconic acid, which will eventually, under the influence of UV light, to be converted to the cis form. Okay, but I'm interested in this portion, which is the glutamic acid portion. Okay, released from the filaggrin is what we call converted to what we call pyroglutamic acid. And that one will be very important, natural moisturizing substance to, of course, the skin, to moisturize the skin. So these are the things that we tend to see. So get to know filaggrin, very important. Even apart from the keratohyalin granules, filaggrin is also very important in, of course, the granular layer, which will eventually get into the corneal layer, which will disintegrate, and eventually the glutamic acid will be very important in forming what we call pyroglutamic acid, which will be a very important natural moisturizing agent. So there we are. What have we seen? We've seen that. So if I'm now, when I started, I moved from down to up. So let me also move from down to up, or you can also move from up to down, because we'll be filling it this way. So let me go similar way. So we say we have stratum basale, that's the deepest layer, consists of about 1 or at most 2 layers of cells. Okay, they are actually the stem cells of the skin, and that is why when they get bent, okay, then what will happen is that you can't produce any epidermis again. You become highly susceptible and you may die actually. So people dying from burns, yes, mainly means that it goes even beyond the basal layer into the dermis component. Then what will happen is that occasionally, or let me say frequently, we see some cells over there which we call them the melanocytes. We will see the melanocyte in the closer view. The melanocyte will tend to have roughly columnar shaped cell with the apical cytoplasmic processes, which will have melanosomes, which are filled with, of course, these kind of melanin that have been produced by them. So that's what we see. Then even apart from that, you may see some other cells, we call them, this kind of dendritic cells, which are actually the macrophages of the cells. Okay, macrophages of the skin. We will see that. Then when they undergo mitotic activity, this one will contain a lot of mitotic figures in there to undergo mitosis to give rise to the next layer, which have some kind of layers of what we call keratinocytes. So all these cells are keratinocytes. They are the main cells. About 90% of these cells are keratinocytes. Okay, so these ones are actually joined by some cell junctions, which we call them desmosomes. So you can see those desmosomes over there, one of those anchoring cell junctions that we find. And that one, we said that this stratum spinosum will contain some spiny or spinous processes, some kind of cytoplasmic process will be emerging from it. Okay, and that one gives it name, spinous layer. But even then, it will begin to contain some kind of granules. Okay, to begin forming some kind of granules over there. Then we go to what we call the layer, which consists of about 3 to 5 layers, the stratum granulosum. Now this time around consists of a lot of granules. Okay, a lot of granules in there, and these granules, yes, apart from even the keratohyalin granules, it also contain filaggrin granules. You've seen what these filaggrin granules are going to do. They will fill actually the cytoplasm, then making it granular. So that's why we call these cells stratum granulosum. Now remember, I told that the stratum spinosum, you may see, we are going to see these kind of Langerhans cells, these Langerhans cells, that macrophages of the skin. Okay, so actually it is this spinous layer that we see, although sometimes we see it in the basal layer. Then consisting of about 20 to 30 layers, actually in thin skin, both thick and thin, then we are going to have the stratum corneum. Although in thick skin, between the stratum corneum and the stratum granulosum, we have about 1 to 2 layers of cells. Okay, being what we call the stratum, or to 2 to 3 layers of cells. Okay, being what we call stratum lucidum. Okay, that clear layer. Okay, which will be coming from the granulosum. That's in thick skin. But in thin skin, just from stratum granulosum, we go to stratum corneum, the horny layer. And this horny layer will consist of about 20 to 30 layers of dead cells, because the keratohyalin, yes, has caused the death, or let me say the pignotic nature of these nuclei eventually dying or disintegrating. And this, mainly it will be filled with keratin. And that's why we describe the skin as being stratified squamous, the epithelium to be stratified squamous, keratinized epithelium. Okay, that's what we see. Now one thing that I want to say over here is that over here in the stratum basale, if there's cancer over there, we call it basal cell carcinoma. It's usually confined. Okay, its prognosis is good. It tends not to spread so much. Okay, so one thing that you have to know is that if it is thin skin, 4 layers: stratum basale, stratum spinosum, stratum granulosum, stratum corneum. But in thick skin, then you have what you call stratum basale, stratum spinosum, stratum granulosum, stratum lucidum, and then stratum corneum. So please get to know these ones. Another thing that I have to tell you is that in thick skin, which are, for instance, in the palm, in the sole of the foot, okay, these ones will be devoid of hair. Okay, as well as even sebaceous glands. So please get to know that one. And as we said the mitotic activities which are going to happen in the basal layer replace the cells in the spinous layer, and then from spinous layer it will go into granular layer, then from granular layer into lucid, and of course into the corneal layer. So it means that the exfoliation activity or the desquamation activity which is happening in the stratum corneum, okay, will be compensated by the mitotic activity in the basal layer. So there we are. We said that exfoliation, okay, or desquamization, which is a natural skin, exfoliation process. Okay, and that one roughly for the cells to move from the basal layer to be exfoliated in the corneal layer, yes, it will take roughly 4 weeks. But sometimes at old age, for instance, the number of days tends to come down to about 75 days. Okay, and what we find is that these cells, okay, will become, they will disintegrate, and of course through some enzyme activity, and of course become exfoliated. So those cells over there, we can conveniently, although they are keratinocytes, because they are in the corneal layer, okay, they don't have nuclei, we call them corneocytes. Okay, corneocytes in the corneal layer. So we said that over time, actually, the intercellular cement becomes denser. So the connection between these cells becomes denser. So for instance, at old age becomes very dense. And what happens is that it makes exfoliation becomes difficult. Okay, so it may take up some time before they get what we call exfoliated. So that is what we have to know. Good. So now, having seen the epidermis, what are the cells that we find in the epidermis? I said that about 90% of the cells are keratinocytes, which I've showed you, which as they move up, okay, from cuboidal polyhedral cells, they become squamous as they get to the apical most cells. Okay, to about 90% of the cells of the epidermis. And their main role is to make what we call keratin. Okay, and it's the same keratin which is going to kill these cells. Okay, very important. And they will be connected just like the keratinocyte that we find in the case of the stratum spinosum. They are connected by what we call desmosomes, those cell junctional complexes. And these ones arise, okay, from the basal layer through mitotic activity in the basal layer. Okay, so that is what we have to know. So now, what is this keratinization we are talking about? Okay, that one is the formation of a layer of dead protective cells filled with keratin. Okay, in the cytoplasm will be filled with keratin. The keratin is going to kill these cells. Okay, and they are more in areas which are exposed. Okay, let me say areas which are subject to aberration, high degree of aberration, and that is why the keratinization is going to take place in, of course, the palm and of course the feet is more than, of course, that on the forehead. But actually, an area like the eye will not have keratin. And for keratinization to take place, it will take roughly up to about a month. Okay, for what we call the cells to move from the stratum germinativum to the stratum corneum. Okay, for complete keratinization. But I told you that for exfoliation, it will take roughly 4 weeks, or sometimes it may reduce to about 75 days. So please get to know these ones. Now, having seen this keratin, then what is the role of this keratin? Okay, yes, keratinization. Okay, and so by so doing, keratinization is also going to confer some kind of protection to these dry areas of the body, and they form integral structural network that make hemidesmosomes and desmosomes, as well as components of the what we call the basement membrane for structural integrity. Then it's also for maintaining spatial relation between the nucleus and cytoplasmic organelles. Okay, keratin is going to do that, and it's also help in transfer of information, okay, or some kind of communication between the nucleus and the cell surface, and of course vice versa. So for cell signaling, okay, keratin is doing that for cell signaling. Remember that it's actually keratin which will make, for instance, the hair very hard. Okay, make, for instance, the nails very hard. Okay, so it confer that kind of structural integrity actually. Now, the other cell that we find, okay, is what we call the melanocyte. And melanocytes are associated with the stratum basalis. Melanocytes will be roughly columnar with this kind of oval or spherical nuclei or nucleus. And then apical, we have a lot of cytoplasmic processes, okay, in there. And apical too, we find these kind of melanosomes. And melanosomes were filled with what we call melanin, that pigment. So that upon release of this melanin, yes, it will get to the areas where we have the keratinocytes, conferring that kind of dark color to the skin. So that is what we call the melanocyte. So if there is cancer of it, we call it melanoma, for instance. Then what we find is that, yes, I told you that it is actually in the basal layer. So we can see that in the basal, you can see that kind of pigment over there. Okay, these ones melanin pigments in the cells of the basal layer, but these are actually the melanocytes, which will make that kind of either dark brown. We see the various kinds of melanin. Okay, so that is what I want you to know, actually concentrated in the basal layer of the epidermis of the skin. So having seen melanin, then what is melanin? Now, melanin is actually made from this kind of amino acid tyrosine. So tyrosine amino acid is actually very important in the manufacture of melanin. But what we find is that actually we have two forms, although we may have a third one, which is actually neuromelanin. We are not talking about that. With respect to the skin, we have two kinds. We have what we call pheomelanin and of course eumelanin. Pheo and of course eumelanin. Now the word eu means true. So true melanin, that's the dark brown or dark, let me say brown black melanin, because we have two kinds. We have those one which is going to be, the brown type, and we have those one which will be black. Okay, that's what you should know. And then for that kind of pheomelanin, you can say red yellow melanin, or that kind of pinkish red melanin. You can see that. That one will be found in individuals with light skin and of course hair. Okay, individuals. So for instance, pheomelanin, you may see it in some people, let me say among the whites and what have you, because that one gives that kind of yellow red appearance. But one thing that I have to be quick to say is that yes, it's not that we are going to find pheomelanin or eumelanin exclusive to an individual. You may have combination of these, but the proportion of more, okay, will tend to, distinguish one individual from the other. Now, one thing is that you see, these kind of, eumelanin, for instance, whether you are white or whatever, we tend to find it around the nipple, the areola of the breast. Okay, we tend to see it over there, even in the hair. Most people tend to see it in the hair. Okay, in the skin of some individual, the blacks. Okay, so one thing we see that for the black melanin, we tend to see it in non-Europeans, and also in some aged Europeans we see black melanin. But for brown melanin, we tend to see it in young Europeans. Okay, brown eumelanin, true melanin. Remember, if you're an albino, you don't have any of these, melanin, molecules. But remember, even the skin color, hair color, whatever, sometimes even, keratin may also play a role in giving that kind of impact to it. So there's some kind of distribution trying to show you the various areas that where you tend to see. So the more darker you are, the more you are going to have this kind of eumelanin. Okay, the more black you are. Then the more you are less black, then you have, let me say, the brown eumelanin. The more yellowish you are, the more you are having this kind of pheomelanin, or the more pinkish red you are. So that is the distribution across the group. So now, having seen keratinocytes and melanin, then what is the distinction between them? We said that about 90%, so majority of the cells, okay, that we find in what we call the epidermis, actual keratinocyte, with very few, melanocytes in the basal layer. Then one thing is that these keratinocytes will be producing keratin, while melanocytes will be producing, melanin. But one thing that you have to know is that when the basal cells differentiate, or when they undergo mitosis, then they give rise to these keratinocytes. But remember that these melanocytes are not actually coming from the basal layer. They're actually coming from neural crest cells. Okay, neural crest cells, which of course, if you visit the video that I've done on the brain anatomy of the brain, I talked about that. So we actually be seeing it again. Neural crest cells, which often refer to it as the fourth gem layer. Then what we are saying is that yes, we've seen the rule. This one produces melanin or whatever. Now, one thing is that the function, okay, is that these melanocytes will serve as physical barrier between the organisms and the external environment. But these ones will be responsible in producing color or imparting color to the individual. So these are the things. Now, apart from that, another cell type that we find, okay, this is also coming from the mesenchyme, from connective tissue, is what we call the Langerhans cells. Now, these Langerhans cells, yes, they actually lack desmosomes. Unlike in keratinocytes, which they were joined by desmosomes, okay, they will lack desmosomes. But actually, what we find is that these are actually macrophages of the skin. So it means they are coming from the blood, from mesenchyme. Okay, and they will be important in what we call antigen presentation. Yes, so that they can be, immune response can actually take place. Okay, and they will actually constitute roughly 2 to 8%. I told you that these Langerhans cells, okay, will be found in what we call stratum spinosum. stratum spinosum, but sometimes you may find some in the, of course, the basal layer, in the basal layer. So stratum spinosum, but sometimes you may see some in the basal layer. But one thing is that they contain some small rods, okay, or flash-shaped granules, and these ones are what we call the Berbeck, granules, okay, named after the person who found it in 1961, Berbeck, granules. Okay, so that is what I want you to know regarding what we call these Langerhans cells. Now, another cell that we find, which is very important as far as sensation to touch is concerned, is known as Merkel cells or Merkel disc. Okay, that one, they have folded nuclei. Okay, they have clear, organelle-rich cytoplasm. Okay, with that kind of peripheral protrusions. Okay, and they are actually going to be found in what we call the basal layer. So please get to know that in the basal layer. So in the basal layer, you are going to find, of course, melanocytes. You find Merkel cells. Yes, these are the areas that are going to find these cells, important cells, in, of course, the basal layer. Okay, so please get to know that. But what is happening is that they are concentrated at the base of what you call retipex. So if you hear of retipex, okay, base of retipex, then think of what you call the Merkel cells. I'm going to show you that. So there we are. These are the cells I'm talking about. These are the Langerhans cells with the cytoplasmic processes. Very important in antigen presentation. Then in, of course, the basal layer. So this one is in the stratum spinosum. Then in this basal layer, we have these Merkel cells. Merkel cells also present with some processes. Okay, clear cytoplasm with this kind of ovoid, this kind of nuclei. Okay, nucleus in there. So that is that one. Very important, as far as touch is concerned. Okay, fine touch is concerned. So these are the Langerhans cells and, of course, Merkel cells. So I believe you found it helpful. We'll look at the other things regarding, of course, the skin. Next time we are going to look at the dermis. I'm very grateful for your time this evening. Have a good night and may God richly bless you.